Hello, thank you for joining us for the webinar today. I'm Lee Pang from AWS, and joining me from the Broad is Ruchi Munshi. Today, we're excited to be talking to you about the release of Cromwell support for AWS. So in this webinar, we're going to first go through a bit of overview of what AWS services you can use in the context of genomics and research. Then we'll get you a brief introduction to Cromwell and Whittle, followed by a description of how Cromwell supports AWS. So hopefully by the end of this webinar, you'll have a good high-level idea of how to run genomics workloads on AWS with Cromwell. So to start, I'd like to highlight how AWS enables scientific research. We have over 130 distinct services, and new stuff is launched almost every day. For the scientific community, our services first help to remove the IT complexity of running scientific computing at scale, accelerating time to science, and two, provide a number of options to make it easier to access data and perform analytics, which overall drive scientific innovation through facilitating collaborations that can span the globe and enabling scientific reproducibility. And the motto on my team here at AWS is it's about science, not servers. With AWS, researchers are doing amazing and computationally challenging things in fields like weather forecasting, molecular modeling, and astrophysics. Today, we're going to talk about genomics, which is the study of the structure and function of DNA at a molecular level. Everyone has a genome, a long sequence of DNA that makes everyone unique. So being able to know what your genome says about you has important impacts in medicine. And over the past 20 years, technological improvements have made reading a whole human genome steadily faster and cheaper to do. We are now at the point where getting your genome sequenced only costs around $1,000. However, this also means that the amount of genomic data is exploding. The data set for a single whole human genome is about 100 gigabytes in size. And worldwide, this amounts to genomic data that's probably on the order of exabytes. For researchers and clinicians working with genomic data, there are a couple of key considerations. First, genomic data sets are big. Depending on the amount of detail, a single genomics experiment can range from tens of gigabytes to several petabytes. Second, converting raw data from a DNA sequencer to scientific insight takes a lot of computational steps, each with their own unique resource requirements. Often that work comes in the form of processing pipelines or workflows. For example, what I'm showing here is the Broad's genome analysis best practices workflow at a high level. Here you can see that there are many steps to align the raw sequence data to a reference, QC the data, and identify genomic changes that scientists and clinicians would use for analyses. So these workflows boil down to a sequence of tools that convert the data in their own special way. Each tool in the workflow is unique. Some can be command line utilities, others could be Python scripts, and even more could be Java or C++ programs. This means that there are a lot of software dependencies to manage. Similarly, each tool has specific computational needs and performance profiles. Some may be compute bound, while others need lots of memory. Also, parts of the pipeline may run quickly, while others more slowly. This adds complexity to how and when a tool in the workflow should run. So the primary challenges with genomic workflows are one, application packaging, and two, workflow orchestration. Knowing that, there are several key technologies and AWS services that anyone building a genomics pipeline should consider. Docker containers and container registries for making software more portable, managed execution with EC2 instances and AWS Batch, event triggering with AWS Lambda and step functions, and storage solutions like Amazon S3 and Elastic Block Store. So knowing all of this, let's take a look at how all of these services and tools come together. So it really starts with a compute node. In AWS, you create one with a Amazon EC2 instance. In effect, it's a Linux server and an Amazon EBS volume as a disk for that server. It's important to note that in the cloud, this compute node isn't permanent. Once you're done using it, you'll discard it. So after that, you'll need a place to store the data that you need and create from the processing steps that you run. You can use Amazon S3 buckets for this. When you create the compute node, you install the tool that you need through various automation methods. And to get to work, you first put your source data set into your S3 bucket. This could be the raw data for a full experiment or data from a previous step of a pipeline. Then you stage only the data you need for the step the tool represents on the compute node. When the processing is done, transfer those results to your S3 bucket for further analyses or for use in other steps of the pipeline. So this is the basic processing pattern that is used in genomics and across fields in scientific computing. So what if we want to run a couple of tools at the same time that have conflicting dependencies? The practical approach here is to encapsulate each tool and their dependencies in Docker containers. This makes each tool more portable and reduces complexity for provisioning the underlying compute node. These containers can be retrieved privately via Amazon Elastic Container Registry or from public registries like Docker Hub. AWS also provides a machine image or an AMI that is optimized for running containerized processes. This comes with Docker services pre-installed and enables monitoring of container processes with CloudWatch. So now, how does one handle the various compute needs for each tool? Simply pick the most ideal EC2 instance type for your compute node. AWS provides a wide spectrum of options, from instances that are ideal for compute-heavy jobs to ones that require a lot of memory or GPU-based processing. So let's move up a level. So how do you get multiple jobs done efficiently using batch processing? Well, first, you can group specific types of compute nodes into compute environments. These can be based either on-demand instances or AWS's spare capacity, or what we like to call spot instances that provide up to 80% cost savings. You can then queue up your tools in a job queue to be run on these compute environments. And so what sits in between these is AWS Batch. AWS Batch is the service that makes this happen easily. It takes tasks off the queue and sends them to the most ideal compute node in the compute environment you've defined. So it's a fully managed service, meaning you don't have to worry about launching and scaling the infrastructure you need. It's fully integrated with AWS, which means batch jobs can work with all other AWS services. And lastly, it's cost efficient. It helps you run jobs in the most efficient and cost-effective manner, spinning up only the resources you need and turning them off when your work is done. So let's talk a little bit more about queues and cost efficiency. In AWS Batch, you can set up multiple job queues. So you can create a high priority queue, like what I'm showing at the top of the slide here, that prefers to run jobs on on-demand instances, and then on Spot for times when there's really high demand. This is a queue you would use if you had time-critical workflows. Otherwise, you can use a default queue, which prefers to run jobs on Spot instances first. Again, giving you those cost savings. So how are multiple tasks orchestrated? In a couple of blog posts, we described how this would be done using serverless technologies like AWS Lambda and AWS Step Functions. In a nutshell, tasks can be triggered by various events like putting data in an S3 bucket and captured by Lambda. Step Functions adds a layer on top of that, a layer of logic to orchestrate what task to execute next. But today we're excited to announce something really awesome. We're excited to announce that we can do this with tools that bioinformaticians and genomics researchers are more familiar with, like the Broad Institute's Cromwell. At this point, I'd like to hand this over to Ruchi to give you more detail about Cromwell and how it runs on AWS.
Thank you, Lee. So I'll just do a quick introduction of myself. My name is Ruchi Munshi. I'm a product manager at the Broad Institute, and specifically I manage two sort of software components, Cromwell and Whittle. And thanks to Lee, you've already had a great introduction and some like sort of sneak peek insights into what Cromwell does in the world of bioinformatics. So I'm just going to jump right into it. So Cromwell, in essence, is a workflow management system that is designed to run scientific pipelines either on-prem or in the cloud. This means that it has the flexibility to run a pipeline against something like a local HPC cluster and also provide the portability to run the exact same analysis onto a cloud platform. So this means that it's an execution engine with a lot of built-in flexibility. And another great thing about Cromwell is that it's really designed to handle a large number of concurrent pipelines. For example, just a few years back, we worked with a group that successfully completed a joint genotyping analysis on 20,000 genomes. And that was a huge milestone for that group and also for us as an engine. The goal of Cromwell is that it's really meant to reduce the complexity around resource management and infrastructure work that's generally required to run a large-scale scientific analysis. And in particular, as Lee mentioned earlier, that as the cost of sequencing has reduced over the years, there has been a giant spike in the amount of genetic data available for analysis. And so the sort of value add for Cromwell is that it really empowers a researcher to easily scale their analysis against all these large scientific data sets available to them. And most importantly, Cromwell isn't necessarily tied to a specific type of domain, but it's really meant to be a versatile interface for running any type of analysis that can be chained as a set of functions with inputs and outputs. And that kind of transitions me into the next bit, which is that Cromwell uses a specific language known as WDL, which stands for the Workflow Description Language. And it's really a human readable, writable language. And it's a language that provides intuitive sort of syntax and flow for packaging multiple steps, or what we call tasks, into an execution graph, what we call a workflow. And that's the analysis that Cromwell knows how to execute as a pipeline. And as of more recently as well, Cromwell has added the functionality to run CWL workflows as well. So it's sort of growing in the various languages that it adopts to. So there's two main ways to run Cromwell. So on the left-hand side, we have an example of sort of a single line command that can be used to run Cromwell, to invoke Cromwell. So that kind of a mode is called what we call run mode. And the advantage of using run mode for running Cromwell is that it's essentially a single command. It requires very little configuration, and it's the perfect kind of way to be able to cycle through the same workflow over and over again as you're developing it, modifying it, without having to sort of leave the terminal at all. And so run mode is often the common way that an independent analyst will sort of prototype their workflow until it hits like a final version. And then another way to run Cromwell is in server mode. And it's different in the sense that while it requires accessing the API endpoints to be able to submit and track workflows, running Cromwell in server mode is much more scalable. And that often means that there is a little more complexity setting up Cromwell in server mode around finding the proper configurations for the kind of work you're trying to run, and also setting up a MySQL database to sort of keep history and sort of track of all the workflows. But it's really appropriate for production environments. And another key benefit of Cromwell in server mode, also in run mode, is that it has the ability it has a functionality we refer to as call caching, which is essentially given a set of inputs in a particular task. If the task has successfully run before, then instead of rerunning that task, Cromwell is smart enough to be able to copy over the existing outputs, reuse the existing outputs without having to rerun. So this is the sort of functionality that's really wonderful for analysts when they have to sort of restart or sort of resume a workflow without having to spend extra resources recomputing things that have already successfully finished before. So it's a really nice added feature that our users really depend on and love. There's all kinds of backends supported for Cromwell. So one way to run Cromwell is just sort of locally on your own machine. So which is really nice when you want to try it out for the first time without investing too much time and infrastructure into getting started. Another way to run Cromwell is to use an HPC cluster. So it supports running jobs on LSF, SunGrid Engine, SGE, Slurm, and quite a few more. Additionally, it also has a GA4GH test compliant backend. And of course, Cromwell supports running workflows on the cloud. So I'm very excited to say today that we've expanded to being able to run workflows on the AWS cloud platform by taking advantage of the AWS Batch API. And in addition, Cromwell is also able to work on other cloud backends like Google and Alibaba and more. So this diagram will probably look familiar to you because it's a piece of what Lee walked you through earlier as well. And it's kind of a zoomed out picture of where Cromwell interacts and kind of sits on the existing AWS stack. So on the right-hand side, you'll see the various different compute environments set up. And you see that AWS Batch is sort of the source feeding in new jobs to the compute environment and starting up new nodes. And then sort of right before, sitting on top of AWS Batch is the Cromwell server itself. And in this case, the Cromwell server, which is distinguished as a little pig, which is our sort of our icon for Cromwell, the engine, is that it's sitting inside of an EC2 instance. The server is running from there. And what the Cromwell server is doing is that it's the one managing the API calls being sent to AWS Batch, and AWS Batch is the one sort of managing the AWS resources. So in every way, I feel Cromwell is sort of the conduit to the cloud platform, and it takes away the burden of resource management and task orchestration away from a user and onto itself, which is what makes it so easy to use and powerful for a researcher who's not necessarily a very like a technical software engineer, but just an analyst trying to get their work done. Okay, this is a high-level overview of the requirements to be able to configure Cromwell for AWS. There are certain sort of base requirements, such as having an S3 bucket set up, which is where all your workflow outputs get gathered. There's a QARN, which would be required as well. And while this is pretty high level, we plan on making sure that there's thorough documents available as a link to this webinar, which will have in-depth steps that are needed to be able to get all these components set up, like the S3 bucket and the queue and any IAM requirements and such. So this diagram will also look familiar. You'll see again sort of the compute node pairing of the EC2 instance and the EBS volume, which is where sort of most of the action happens, I feel, which is where an actual analysis is run. And then on the left and right hand side is sort of like the S3 bucket, which is what holds the state of the data being analyzed. Here are the inputs coming in, being staged onto the machine, and then once they've been transformed, then the outputs being staged back out. And in the case of Cromwell, we have a custom AMI image that's used. That's really just optimized for the case of how we've historically seen users of Cromwell run their jobs. And there's sort of two big advantages here that are worth noting, which is that in our case, since genomics data is very, very big, when using Cromwell with AWS Batch, what we do is we ensure that there's always always enough disk space available, and we do that by sort of monitoring and auto-scaling the disk requirements as an analysis is actually running, which is a really nice way for a user to not have to worry about managing disk requirements and knowing that it's been taken care of. And additionally, there's utilities available to be able to do more in-depth logging and more in-depth monitoring of what's happening inside of that task. So there's ways to use CloudWatch with your logs and just leave all the data staging of gathering inputs and staging outputs back fully to Cromwell. Okay. So here's a quick overview of WDL, the actual workflow description language that Cromwell runs. And I'm just going to go through some high-level concepts that are worth noting. So essentially, a workflow is composed of various different tasks. And you can sort of think of a task as a function. And on the right-hand side, we have a stanza for a task named Task A. And in the yellow, you have highlighted some inputs being passed, inputs that are required for that task, a few different file objects, a string object, and then directly below it is a command, which is the actual command line tool or the invocation of an analysis that will take place. And you'll see in that command itself, one can reference all the inputs of that task as well using some specialized syntax. And then additionally, when running Cromwell in the cloud, for all cloud backends, Docker is a very important requirement because that's how a user will encapsulate all their software dependencies sort of in an easy and portable way. And so WDL has a syntax needed that on a per task basis, one can change the Docker image associated, sort of required almost to run their command. And then, of course, there's syntax below it to gather sort of the expected outputs that are interesting to a user. And specifically, these are the outputs that one would want to capture and store or possibly chain and pass along to a downstream task. So on the left-hand side, we see two calls being made. There's a call for task A, which is sort of an execution, which sort of invokes the execution of task A. And then directly below, call task B, which is an invocation of executing a different task called task B. And you'll notice that one of the inputs for task B is the output from task A. And so there's very simple syntax to be able to associate the chaining between the two tasks. And just the way one passes in inputs for a task, it's the same for a workflow. And those inputs can be easily referenced as well as task inputs or used wherever needed. And usually a workflow file contains the tasks being run by that workflow and just the chaining of the tasks inside the workflow definition directly. So we talked about this a little bit, but the runtime parameters for inside of a task are very important because they sort of dictate the resource requirements for a particular task. So for example, Cromwell requires that there be sort of a container solution for running in the cloud. And so Docker, so in this particular case, we see that an Ubuntu Docker image is what's being used to run the command. There's ways to specify if there's different disk requirements and of course different memory requirements because like Lee mentioned earlier, not all tasks will be equal. So we want to be able to allow people to sort of define the shape of their task as needed per task instead of sort of standardizing it for a workflow. So this just provides really nice flexibility in terms of resource management, and specifically because by being so specific around resource management, that's a really easy way to manage cost for your analysis. And so the runtime block is really about sort of like time and cost optimizations that a pipeline author can choose to make for their particular analysis. So just a recap for all the things to keep in mind for trying out Cromwell on AWS is that you'll need to grab an EC2 instance on which to run the Cromwell server. there's an optimized AMI which is used, which has some customizations, particularly keeping in mind genomic workloads and just sort of the general requirements for interfacing with Cromwell. You'll need an S3 bucket setup, which is sort of the place where all your Cromwell outputs will be deposited. And additionally, you'll need to enable the AWS Batch API, which handles the compute environment and job queue components of this orchestration. So the main benefit that we provide through sort of this entire stack is that Cromwell and WDL make it very easy to write and express complex genomic workflows using the WDL language, and by providing a very straightforward interface to run that analysis. And Cromwell and AWS is really meant to be an avenue or sort of an option for users to be able to plug into this enormous, the enormous like AWS cloud platform, and to be able to scale much more seamlessly than they can through their on-prem infrastructure. And if you're interested in learning more, here's some links to some docs. Cromwell is fully an open-sourced, is a fully open-sourced repository, so you can see all our activity and sort of plans in flight. And WDL is the same way. It's sort of an open effort where we take in active contributions, and we love getting feedback on both our products. So feel free to drop us a note. And in the future, you'll also be able to see more docs and sort of tutorials on how to get started. And there's just so much more reading around sort of the various different components of AWS as well. And I'll hand it back to Lee.
Thanks, Ruchi. As we wrap up here, I wanted to highlight some additional resources for researchers interested in AWS. So if you want to learn more about what other researchers are doing on AWS, we have a lot of case studies and examples on our page for research and technical computing. Next, I suggest you check out our Research Cloud program. Signing up for this program gets you a researcher's AWS handbook, which we've heard from the field as being dubbed the missing manual for doing science on AWS. And if you need help for an innovative idea, we also provide credits to help you get started. Last, if you need data to test out Cromwell and AWS, I recommend you look at our registry of open data. We have several genomics data sets that you can try out there. So I want to thank everyone for watching this webinar. I hope you are as excited by this announcement as we are. We're really excited to see what you in the research community can build with this. And to help you out, we're offering $250 in credits to the first 100 participants to register on our launch page. So be sure to use this code that you see here to let us know that you are in this webinar. And finally, if you do have any questions, we are hosting office hours today from 2 to 3 p.m. Eastern Time. Join us at the meeting link below. And that's all we got. Thanks, everyone, and thank you, Ruchi.